biology students. Today we're going to start our unit on molecular biology, and we're going to start with lesson 1, which is called the discovery of DNA, its evidence, and the structure. So you are going to need the note taker that looks like this. As you notice, you have a list of scientists that made important contributions to the discovery of DNA. So next to each scientist's name, you're going to write important information that you hear throughout this video. Okay, so let's get started. Okay, so first we need to start with the location of DNA. So in previous units, we learned that cells can be prokaryotic cells, meaning they have no nucleus, or they can be eukaryotic cells like you, and they do have a nucleus. So as you notice here, inside the nucleus, we have the DNA. Now, DNA can be packaged in different ways. So we see that one way DNA can be packaged is in a chromosome formation. And notice that the DNA is very tightly wound up. And you kind of see like this X structure here of a chromosome. Now remember that you will only see chromosomes during mitosis, and to be more specifically during metaphase of mitosis. But if we were to zoom in on this chromosome, we would be able to see the molecular shape of DNA. And the molecular shape of DNA that was discovered is a double helical structure. So the backbone here in gray is the phosphate sugar backbone, and inside with the colors here, with the red and the yellow and the green and the blue, we see the nitrogen bases, ATGC. So here we start with the historical outline of the discovery of DNA, and this is where you're going to start taking some notes. Starting in the 1860s, when DNA was actually discovered, and then moving on all the way to the 1950s, where the structure of DNA was discovered. So first we're going to start with Gregor Mendel. So you learned during the genetics unit that back in 1866, before many other significant discoveries and findings were made, Mendel actually discovered that there was something, that there was an agent that was being passed down from generation to generation. Now, he didn't really know what that agent or what that something was, but he knew that a parent could pass on something to the offspring, and that's how characteristics and traits were passed down. It wasn't until 1869 that Friedrich Miescher, he began working with white blood cells, and he started working with the pus within the white blood cells, actually, that he collected from bandages. He would manipulate that pus by adding a weak alkaline solution to the pus, which caused the cells to decompose and the nucleus to move out of the solution. So he isolated that nucleus, that nuclei, and he called it nuclein. Now, he didn't realize that what he was looking at was actually DNA, but he was the first one to actually isolate the substance that we now know as DNA. It wasn't until 1881, a few years later, that Albrecht Kossel, he won the Nobel Prize, and he actually named and coined the term DNA after identifying it as a nucleic acid and isolating the nitrogen bases. So we know that DNA is one of the two nucleic acids, which are the big macromolecules. So moving on, now we're in 1928. Scientists want to know what the transforming agent is that actually carries the genetic information from parent to offspring. So there was a debate at the time as to what macromolecule carried the genetic code. Was it DNA, being a very simple molecule, only made up of 4 nitrogen bases, or was it protein, a more complex molecule actually made up of a combination of 20 amino acids? So that debate ensued, and there were a lot of scientists who believed that Protein indeed was the macromolecule that carried the genetic code because it was complex, right? That made sense. Um, no way DNA, being as simple as it was, could carry the genetic code, right? So in 1928, Frederick Griffith, he conducted transformation experiments in rats, and he noticed through experiments that there was indeed a transforming agent that was passed on from a parent to an offspring. That transforming agent um, carried a disease actually that would kill off his rats. So he wanted to know if the transforming agent was DNA or proteins. He wasn't able to determine what it was, um, but he is given credit with actually determining that indeed there was a transforming agent passing on genetic information from parent to offspring. It wasn't in a period of about 20 years from the 1920s to the 1940s that many scientists actually were experimenting, trying to find out what was Griffith's transforming agent. So Avery, McLeod, and McCarty did lots of experimentation and conducted lots of and collected lots of data, and they came up with the conclusion that it was probably DNA and not proteins that was the transforming agent. They didn't have enough data to convince the entire scientific community, so their evidence was not widely accepted by other scientists. But it wasn't until 1952 that the technology got a little bit better, and Alfred Hershey and Martha Chase, working with bacteriophages, actually experimentally concluded that DNA is the transforming agent and therefore the unit of heredity. And it was through their experimentation that the entire scientific community agreed that DNA carried the genetic material and not protein. So the debate was settled once and for all. Now, we also need to understand the contribution of another scientist. His name is Erwin Chargaff, and he discovered that DNA varies between species. And he came up with what we now know as Chargaffs Rules. And this proved that guanine and cytosine units, as well as adenine and thymine units, were the same in double-stranded DNA. So what we understand today, being a Chargaffs rule, is that the number of adenine, or the number of A, is equal to the number of thymine, or the number of T's, and that the number of guanines, or G's, is equal to the number of cytosines, or C's. So now scientists very well understood that DNA is a transforming agent in Griffith's experiments, and they understood that DNA, even though it looked like a very simple molecule, right, with only 4 nitrogen bases, is the one that is responsible for transmitting the hereditary information. So again, here is Chargaffs rules, right? A is equal to T and C is equal to G. And if we look at Chargaff's discovery a little bit in more detail, we know that he was a biochemist. So he studied the composition of these nitrogen bases in a little bit more detail. And what he noticed is that the adenine base and the guanine base, they resembled each other biochemically. So they were much larger bases than the pyrimidines, that T and C. So he called these bases T and C pyrimidines, and he called A and G purines. So you can also say that in DNA, the number of purines is equal to the number of pyrimidines, because A's are equal to T and G's are equal to C. So we can also say that the base pairs are complementary. So that's what we now know as Chargaffs rule. And the way that I have you guys remember is apples in the tree, A is equal to T, and car in the garage, C is equal to G. All right, so once scientists confirmed that DNA carried the genetic material, they wanted to know what the structure of it was. And the race began. So here you see a picture of the scientists who discovered the structure of DNA back in 1953. We have James Watson and Francis Crick, we have Maurice Wilkins, and we have Rosalind Franklin. And then we have this very famous picture called Photo 51, which is an X-ray diffraction picture that Rosalind Franklin took. And we'll learn in just a moment what the importance of Photo 51 was in the discovery of DNA. So let's start with Franklin and Wilkins. So in 1951, Rosalind Franklin's work in X-ray crystallography began when she started taking X-ray diffraction photographs of DNA. So she was an expert crystallographer. Her images, and more specifically Photo 51, showed the helical form, which was confirmed by Watson and Crick nearly 2 years later. Her findings were only acknowledged posthumously. That means after she died. Maurice Wilkins, now he worked with Rosalind Franklin in the same laboratory at King's College. He also began studying nucleic acids and proteins via X-ray imaging. He was very successful in isolating single fibers of DNA and had already gathered some data about nucleic acid structure when Rosalind Franklin, who was the expert in X-ray crystallography, joined the unit. Now, there was much tension between the two of them. It was also rumored that they had a romantic relationship and that they didn't see eye to eye and lots of things. And some people might even say that if Wilkins and Franklin had cooperated better, they might have been the first ones to discover the structure of DNA. Indeed, much of Watson and Crick's model was based on photographs taken by Franklin and used without her permission. So Photo 51 that I showed you before, this picture, was taken by Rosalind Franklin, and then Maurice Wilkins took Photo 51 and showed it to Watson and Crick when they themselves were trying to figure out the structure and shape of DNA. Now, before Watson and Crick looked at Photo 51, they had the shape of DNA kind of inverted. They thought that the backbone, which is the phosphate sugar backbone, they thought that was in the interior of the helical model, and they thought the bases were on the outside. And they were incorrect, and it wasn't until they saw Photo 51 that they realized that the bases must be on the interior and the phosphate sugar backbone on the exterior. So in 1953, Watson and Crick published that DNA structure was indeed a double helix structure, formed twists to form a ladder, was made up of 4 nitrogen bases inside, A, T, G, and C, and had a sugar phosphate backbone on the outside. So Watson and Crick were given credit for the discovery of the structure of DNA. So Maurice Wilkins shared the 1962 Nobel Prize in Physiology and Medicine with James Watson and Francis Crick. And notice how Rosalind Franklin was not given the Nobel Prize because she had passed away at that time. So there we have it. Here we see the DNA structure. It is a helical structure, and if you were to untwist that helix, you were to see the bases in the inside, A, G, TC, the sugar phosphate backbone on the outside, and indeed a very beautiful model.